who wasn't able to join.
Perfect. Well, first, I'd like to welcome everybody and just introduce myself. My name is Rachel Long, and I will be taking over the hosting of this N3C Community Forum from Dr. Joel Saltz. I think we really owe him a lot of gratitude and a big thank you for doing it all these years, but he's decided to hand over the reins, and here I am, so I will try my best. So welcome, everybody, and Happy New Year. And next slide, please. And just as always, I want to mention that the presentation will be recorded and it can be found on the Data to Health YouTube channel. And for any questions that you have, just please feel free to post them in the chat. We'll be monitoring it. And if you'd like to say some words, just go ahead and raise your hand, and Tersine will help so that you can unmute and ask your questions. Next slide, please. I'd like to have a brief call out that with the current state of the enclave, we are now up to 8.5 million COVID cases and about 2.8 billion clinical observations. So we have quite a bit of data, up to 83 sites as well. Next slide, please. And for the next upcoming forum presentations, we're going to have a couple of great speakers lined up for you. There is on January 22nd, we're going to be learning about machine learning-based phenotype for long COVID in children. And then on February 12th, we're going to be talking about AI functionalities in the enclave. Next slide, please. And I want to introduce the speakers for today. We have Dr. Chris Chute and Dr. Richard Moffitt from Johns Hopkins, and now Emory as an update to this slide. They're going to be talking to us about the N3C tenant model, and I know we're all interested to hear about this. So I'll turn it over to both of you.
Ken has his hand up. Ken?
Oh.
Yeah, thanks, Chris. I just wanted to, just hot off the press and want to take advantage of the opportunity. We just received the SEER data set for all of N3C COVID. As of an hour ago, it was linked and it's actually in the enclave. And I just wanted to ask your help. I'll be sending out a letter to the sites because we need to get your permission to allow us to link it to your specific clinical data. So I just wanted to ask your permission to do that, and more to come. Thanks, Chris. Sorry.
Well, that's outstanding news, Ken, and thank you really for all your heroic efforts in negotiating these data linkage opportunities. They help enormously. Of course, I'm upset that you took an hour to tell us, but that's all right. We'll get over that. Thank you for that kind introduction, Rachel, and welcome everybody. First slide, please. We're going to, Richard and I, talk a little bit today about the who's showing the slides? Should I be doing?
Yeah, sorry, Dr. Chute. The slides weren't shared with us.
Oh,
I'm
they're
happy to
secret.
share
I
them.
see.
Yeah,
no, I'll go ahead and do it. Forgive me. Let's see. We want to go to the first slide. I'm sorry. Well, Richard and I are going to talk about what's going on with N3C from here on out, the N3C beyond COVID, which is an update on what we did over the past year in what's widely known as the tenant pilot. Next slide. Oh, I am the next slide. So if you think about it, there are many kinds of consortial efforts to bring together health information and data. And I think we all recognize that it is inevitable and unavoidable to integrate data across academic centers and other centers, because the kinds of questions we're trying to ask, precision medicine issues, detailed issues, machine learning issues, we need that amount of data. So integrating and harmonizing data enhances the power, reduces bias, obviously rare disease and niche cohorts. It's inevitable. The question that arises is who's going to do that? We know that insurance companies are doing that without any effort to make it shareable or open or public. EHR vendors are doing the same thing. high-tech companies, the Googles and Microsofts of the world. And of course, it's our aspiration that it would be the academic community that would ultimately lead these efforts, really for the public good and for our shared opportunity for discovery and analysis. We assert that we think N3C was pretty successful, no matter how you measure it, volume of records, investigators, sites, publications, but that machinery can be reused and is arguably disease agnostic. There's nothing COVID specific about the infrastructure that was built. We have established synergy of a lot of federated data repositories from contributing sites. The phenotype queries and scripts are completely transformable and can be used to ingest the data for any purpose. The multiple the common data models that we've supported to OMOP transformations will work, whether it's COVID or not. The FedRAMP secure environment repository, of course, exists with the analytic environment. And N3C Clinical seeks to leverage that infrastructure beyond COVID, and we did a pilot in 2023. There are things that are very similar about it. Continue to leverage the existing networks, the federated networks, which really do most of the data curation, if we're to be honest. Continue to provide executable queries and all the common data models that enable that data acquisition, centralization and harmonization of the data, a highly secure FISMA moderate compliant data environment, and to operate as a federally managed cloud, which has great advantages for data linkage, as I think Ken just mentioned in the beginning of the call. We have the historical shared governance of the community, of NIH and NCATS, with shared responsibilities for a number of activities, as you can see, and then community-focused activities and NIH-focused activities. NIH remains the fiduciary agent and holder of the data, which we think is appropriate, and thus owns the Data Access Committee as they're required to do under federal guidelines. The governance overview, we still have a single IRB in the pilot that we had and presumably going forward. Richard.
Yeah, so the differences between N3C, what we're calling N3C Clinical for the clinical pilot, and N3C COVID are that during the pilot phase, there were lots of cost controls imposed. So as opposed to the COVID enclave, where there's 83 sites providing data, and currently, as of the last time I checked, there were 537 active projects. For the pilot phase, there were only 10 data providers sending data and only 3 projects authorized. There were completely new central IRBs and NIH IRBs governing this, completely new data transfer agreements governing the data coming into the pilot, completely new data use agreements for those using the data. And importantly, there is no data transfer or reuse between N3C, which is COVID specific, and the N3C clinical pilot. Important to this pilot phase, which is a little bit different from the way the data is handled in N3C, is that data providers retain agency over the use of their data. Next slide. What we mean by that is that all institutions, they're agreeing to have their data harmonized to a common data model like OMOP. They're agreeing for centralized but discrete data quality benchmarking. What we mean by this is we will continue to give the one site versus all other sites comparison so that sites can learn from quality disparities. And what that implicitly means is that other sites are agreeing to be used in the pool of benchmarks for each other. Site information is mixed for unit harmonization purposes, so trying to figure out units of measure when they're missing. And then there's limited reuse of the data for high-level statistics, for example, dashboards, in a way that prevents re-identification of the sites. So for those of you familiar with the concept set browser, that we're implicitly sharing data across sites there to show term usage, patient counts for diseases, et cetera. So these are the pieces of reuse that sites are sort of blanket agreeing to by participating in the tenant model. Next slide.
I don't mean to interrupt. I was just there was a comment that the top part of the slide's not showing. I was wondering if
Yeah,
on your
Chris,
end.
your Zoom, the very top bar might be obscuring. I don't know if you can get that somewhere.
Yeah, if not, that's
I'll
okay.
work on trying to move it. I'm not sure how to do that.
Okay.
It's really just showing the top bar of our titles. Anyway, so N3C clinical data contributors retaining agency over their data.
Oops, sorry.
Unlike, there you go. That worked, but now it's in the
Good.
bottom right, which is
Oh.
possibly
Even worse.
less critical. Anyway, the sites do retain agency over the use of their data for research. So sites can opt in to any project, which is different from N3C, which is a blanket agreement for any COVID-related research that's approved. So this could enable at least these three modes that I've outlined here. You could have a large pan-CTSA project that resembles a COVID enclave. For example, if every site opted into a COVID tenant. You can also have smaller projects with limited participation and sensitive linkage. The example comes up of looking at drug addiction and getting data from Betty Ford clinics. You can also have domain-funded research with enhanced linkage. As Ken just linked, maybe NCI wants to sponsor something and include links to the SEER database. For everything going forward, contributing sites will be able to choose limiting their participation to these sort of defined categories. So you could say, I want to participate in anything that is NIDDK funded research. For example, a site could opt in and say they agree to participate in all projects approved by the community, which is the blanket reuse case, or they can choose on a project by project basis to participate or not participate. And when I say participate, I mean, will their data be able to be used and provisioned for that research use? Next slide, please. So in contrast to N3C COVID, where we're pulling out patients that are COVID positive through what we call the N3C phenotype, here in the pilot, we're using a more longitudinal phenotype, which is much more permissive and inclusive of patient data. So the goal is to identify patients who receive regular care over a longitudinal period. And so the bar for being included in the tenant pilot was the patient had to have a blood pressure or a body weight measure repeated at least once over a time period that spanned more than 2 years. So you can imagine that's going to catch most patients that occur within an EHR system. So the same as N3C is that this phenotype and extract process happens based on scripts that are posted on GitHub. So we try and make it as easy as possible, the same way that enabled 80 plus sites to contribute to N3C. They can extract with Python, R, or SQL, and all the structures and the folders are the same, and the SFTP credentials were the same. So we tried to make it as light a lift as possible for these sites in the pilot, and there's no reason that it's going to be any harder going forward. So what's different is it is not disease specific, right? There is no mention of COVID or diabetes or Alzheimer's anywhere in the phenotype. It's just, do you have a blood pressure and a body weight? Also, there's no case control matches, which is one of the most expensive pieces of logic included in the original N3C COVID phenotype. And then payloads are not automatically combined and released for research like they are in N3C. They still go to a centralized place for harmonization, and then sites have to opt in to different tenants for research use. Next slide, please. So as you can imagine, governance of this project sort of evolved over time. So we've had these meetings that happened on Friday mornings at 10 Eastern, where representatives from multiple different sites, including all the data providers, as well as members of the community from N3C to meet and codify and make changes to the governance as the pilot phase evolved. So one thing that got defined, including which global data uses were acceptable. I outlined some of those earlier, like was it acceptable to use the data for dashboards, for example? And then another large hurdle of governance we overcame was how to allow analysts from non-contributing sites to participate. the way the rules were written at the start, if your site participates in a tenant, then researchers from your site are allowed to actually go access the data, and no one from a non-participating site is allowed to, at the very beginning, go in and participate and analyze that data. But we added in a mechanism by which the sites that are data contributors to a tenant can vote to allow members in from outside systems. Data access proposals, which are basically just project choices. We ended up with 3 tenants for the pilot phase. So NIH generously funded enough resources to have 3 projects going. And the selection of those projects was reviewed and approved by the community before they were selected and before they were spun up, with input from all the data contributing organizations, because those data contributing organizations themselves had to opt in. And so we wanted to get broad spread coverage from all, or at least interest from those data contributing sites. Next slide is back to you, Chris. Chris, you're muted.
Sorry about that. It's important to recognize that the N3C clinical infrastructure was leveraged from what we had done with COVID. It was a complete reuse of that infrastructure. And for example, if we look at the communications infrastructure, the data repository infrastructure, the analytic pipelines, the GovCloud environment, the logon options, the multi-factorial validation and authentication, it was all reused and repurposed, as was the harmonization pipeline, which is perhaps the most important aspect and the most costly aspect that was generated in N3C COVID. The governance was completely, well, it wasn't completely the same. We did leverage most of the governance, but we added this component, as Richard said, of the agency of the sites that contributed over the use of their data. That was very different, and I can't emphasize that strongly enough. COVID was a pandemic, it was an emergency, and I think people and organizations were comfortable with the idea that we had to do analysis in a timely basis. But N3C Clinical is not based on a pandemic, and therefore the retention of agency by contributing organizations is very important. We had, as I said, the pipeline that had scalable harmonization with monitoring and provenance. We did over 50,000 transforms on 232 sites that involved 2 million semantic transforms, actually. So all that machinery for pipeline versioning, deployment, and upgrade was reused and repurposed. And I think the pipeline could be refreshed and run in 20 minutes. It was a well-oiled machine that remains very well-oiled and functioning as a practical asset. This, of course, is Richard's work. We did a lot of data quality enhancement in terms of, for example, inferring units of measure where they were missing. Children measured in grams or ounces, and adults measured in pounds or kilograms. Those conversions are trivial if you know what they are, but if you don't know what they are, then with this example of no unit of measure known, we relied on the envelope of distributional parameters to infer what the units of measure were. And you say, okay, that's no big deal, because how often were the units of measure missing? And if you look at this graph, all that blue data was recovered and salvaged effectively using these unit of measure inferencing techniques. You may not be able to read this, but it's pH, CD4 over CDB ratios, BUN creatinine, BMI, respiratory rate, ALT, AST, and so on. Lots of variables that had no units of measure that we were able to leverage in this enhancement purpose. Additionally, as Ken mentioned, there's a lot of linked data, not the least of which is social determinants of health. All of the machinery, expanding data collection, publicly available data sets, the way we managed American Indian and Alaska Native within the environment was all repurposed from N3C and enabled to N3C COVID and enabled to be used within the environment. We also know that the EHR data by itself is not sufficient. We had the opportunity for data enhancement, not only with social determinants, but CMS claims data, which gives a much more comprehensive picture of what's actually happening with a patient. Viral variant data, That was fairly specific to COVID, but the opportunity to do so with other diseases remains possible. Future pandemics, for example, mortality data, the clinical data, vaccine data, imaging data, and so on, with privacy-preserving record linkage. And again, that machinery was brought forward to N3C Clinical. The privacy-preserving record linkage validation, this is an example of where we matched the cohort against CMS data with very, very successful results and very small amounts of discord in the data that we actually used for matching. Back to you, Richard.
So of the 10 sites that were invited to participate in the pilot, 8 of them ended up contributing to all 3 of the tenants. One of them contributed just 2 of the tenants, which is at least a proof of concept for participation. And then one site ended up not contributing to any of the tenants in the end. That site was a pediatric site, and after we ended up choosing all the projects, none of them were particularly of interest for pediatric patients. Next slide. So the total patients that were able to be ingested using the generic phenotype was around 5 million patients from those 9 sites. And each of those got filtered further for a tenant-specific phenotype. And we ended up with 19,000 patients in the Alzheimer's tenant, much more permissive group, 3 million in renal, and 250,000 in the pulmonary tenant. And for the purpose of the pilot phase, we only used level 2 data, which did not include exact locations or exact dates. Next slide. So this big diagram here is a overview of what we've been trying to explain this time as a picture. So the data coming in from the site are coming in from the sites and the FTP at the top. They come to this sort of repository of all data where all the harmonization takes place. And then phenotype extraction scripts pull that data, but only specific chunks of it that are relative for the research that tenant is set out to do. Not only are we pulling out patients based on characteristics, but we're pulling out data only from sites that are opting into each of those tenants. Now, something we haven't really talked about so far, but there is a way that we could go left on this diagram to basically return data to the sites that have been harmonized into the OMOP data model, including all of the bells and whistles that we add on, notably unit harmonization. So your data from the site can come in, and let's have Hopkins, for example. Hopkins data can be OMOPified, units harmonization, and returned back to Hopkins for local analysis, or Hopkins could opt into, say, the renal tenant and the Alzheimer's tenant, and their data would only flow to those 2 tenants. Their researchers would be able to work in those 2 tenants, and they would have to be voted in for a 3rd tenant that they did not opt into. Next slide, please. So the 3 tenants we did in the pilot phase were Alzheimer's, renal, and pulmonary. We anticipate in the post-pilot phase to continue the renal tenant. There's support for a cancer tenant already secured, support for a tenant dedicated to education, which will use completely de-identified synthetic data, and Ken and NCATS is diligently working on establishing more research domain specific tenants. Next slide. So just generally status and timing of the whole project. So as those that have read the fine print in N3C, the existing data transfer agreements cover 5 years of use. Whereas in the clinical pilot that we just did, data access expires next summer. So we finished the pilot phase last summer, and the data is good for 1 more year after that, in which it must be destroyed. Going forward into what we're calling N3C Clinical, NCATS is currently revisiting and revising that data transfer agreement and data use agreements in response to lessons learned from the pilot to hopefully establish these transfers and these data flows going forward more permanently. Currently, NCATS is gauging community support and interest to inform the scope in terms of the number of tenants and the number of users. So if you're interested in this, we encourage you to contact your CTSA head to note that you want to participate in this project going forward. And as I said earlier, NCATS is working to build cross-institutional partnerships, for example, gaining support from NCI to help sponsor a cancer-related tenant and NIDDK to work together on establishing a renal tenant, etc. Next slide. So the next couple of slides here are going to be a diagram showcasing sort of our vision of what this N3C clinical environment can be and could be, and the flexibility this tenant model affords us. So right here is essentially how N3C works. We have data from sites coming in. The COVID phenotype is applied to them before the data comes into N3C. So only COVID-related patients come in. Through PPRL, we're able to link data from commercial entities like obituary data for mortality, for example, or registry data coming in can be linked through PPRL. Also, public data is pulled in, and all of these things are combined into N3C. All the data that's in N3C is available for researchers to pull off pieces of it to work based off of data use requests. So each of those tiny little purple boxes coming off represent the multiple different DURs that you're all used to establishing in N3C, and we're all pulling off the central piece. One slide forward, please. So if this were to work in the new clinical model, the changes would be that the data from sites comes in a much more permissive manner. We're getting basically all the patients from those sites with blood pressures coming forward. That data goes into the main repository, which we're rebranding as N3C Clinical. And if we were to establish a COVID tenant, a COVID phenotype would be applied, and that's where we'd pull out all the patients, for example, with a COVID diagnosis and a matched negative control. At that point, only the sites that opted into the COVID tenant, all of their COVID patients would be put into a COVID tenant, and then all of the users coming in would be pulling off data use requests off of that COVID tenant. Now the arrow comes around, which is really the new innovation of N3C Clinical, is that now each site has agency over whether their data will flow through to that tenant. Also, the data providers that are not the sites would have similar agency. So perhaps we were only able to secure licensing, hypothetically, for SEER data to go into a cancer tenant, then they would have the ability to only opt into a cancer tenant going forward. Next slide. So with this flexibility comes the ability to establish all kinds of different tenants with different scopes and purposes. So here we have at the top a cancer tenant, which might exist, and then an R01 funded researcher from NTI might be working inside the cancer tenant. We might have a NIDDK sponsored researcher establishing a data use request that is working under the guise of a renal tenant. We might have an education tenant, which is less than hypothetical at this point, which is great, where maybe data coming from sites goes to the educational tenant. Maybe only synthetic data comes into that education tenant, depending on what the sites themselves want to do. We could establish a COVID tenant, and it could look exactly like N3C as far as users are concerned. We might be able to establish individual tenants for individual sites to work just on their own data. And it's even possible that commercially funded tenants, like the bottom one down there, might be established. And sites could opt in depending on the purpose and the phenotype and the governance for that tenant. We might be able to link specific study data to specific tenants. And generally what I'm trying to show here is that there's really a large flexibility to be able to reuse the centralized harmonization architecture on the left only one time and be able to achieve and serve all these different research use cases which may come into being. Next slide. I think it's back to you, Chris. Yep.
Thank you. So we learned a lot of lessons in the pilot. The pilot, in terms of data aggregation and integration, was a one-year period, and then that has ended as of last August. What we've learned is that governance, which we sort of knew, but it was reaffirmed, is a team sport. We think the success of N3C was really due to its shared governance and its implementation in an enclave, which gave great comfort to IRBs and organizations around the country. The pilot built on this, but has a lot more potential for data sharing agency and access control by the data providing organizations, which I think we've described. The reusable artifacts and collaboration are critical. We had lots of artifacts. We had value sets and data transforms and coding logic and other kinds of things that were completely reusable. And the sharing promotes collaboration and attribution, which I think N3C received a lot of credit for in terms of giving attribution where it was earned, and that can be done across tenants as well in the N3C clinical. It enables quality research, or enabling quality research really requires a lot of things. Understanding the provenance of the data, where the data came from, designing the right observational study and the best practices that can be done on a tenant by tenant basis, and enhancing the data through data linkage, which again can be done selectively for some specific sources. For example, Richard often talks about the renal transplant data potentially being linked. Obviously, that would pertain to the renal tenant, and it need not necessarily be accessed by other tenants, so that data linkage can even have agency, as Richard characterized. The timelines we had, now this is back in 2022, I guess, so 23, where we had the agreements for review, the sites executed, first data agreements, collaborators, and then in August of 2023, we hit the project milestone where we were able to demonstrate the integration and use of the data. And we're now at a period where we're in the analysis of the data. Joanna said today that I think it's the Alzheimer's tenant and the COPD tenant are now available for projects and analysis among users and contributors. We're ending this chapter of the COVID chapter. COVID is arguably no longer a pandemic. We've run the race. N3C COVID will discontinue onboarding new sites for COVID only as of March 1st. And what that means is that sites that are already working on a data transfer agreement or the IRB may complete the process if they start it before March 1st. But we will not support sites beginning the DTA or IRB process after March 1st of this year. NCATS is considering new data transfer agreements to extend the analytic access to the existing COVID data. We don't want to throw all that data away. And we get queries several a week, actually, of people that are submitting R01s or R23s or various types of grants that want to use the COVID data. And of course, NCATS and the CTSA community want to support that. It's very important to all of us. So how we do that beyond the existing data transfer agreement is something that is being seriously explored. I have confidence that a mechanism will be established, but the intention is to make them tenants within the N3C clinical environment with appropriate approval of the contributing organizations who retain agency. We've had a lot of impact. There are over 3,000 citations as of this morning, 3338, with an H-index of 28 for work that's come out of the N3C COVID. It's been a very successful project. There are over 250 papers cited in Google Scholar presently. And we strongly believe that we can have a similar impact for any other disease we might want to by leveraging this infrastructure and machinery, engaging the community, engaging the CTSA community and our partners and colleagues, the IDEA networks and other related communities and organizations to have similar impact in other diseases. It wasn't just publications. N3C COVID influenced White House policy for things like Paxlovid purchasing and acquisition. It influenced CDC guidelines. It's had impact across many states in terms of policy. And we know that the importance of real-world data and observational data in modern translational research enables great opportunity that can be achieved across multiple organizations. So lots of things worked. We want to say thank you. I'll spare you reading this entire slide, but I think the message is that the users and contributors were variously pleased and excited to work with what we had done. So I'll stop sharing at this point.
I think there's 4 questions in the Q&A. I think we can knock out pretty fast. I'll start with the question, what is the age range of N3C clinical data? And that is all people. There's no age restriction on the data coming in there.
So we have pediatric as well as adult populations. Right. The pediatric sites didn't want to contribute because of volume, frankly, to the projects that we were doing, and they tended to be adult level projects. So it's not that the data wasn't there, but it was the domain topic.
Okay. The next question, it's kind of easy, is what type of clinical conditions data is collected? Is it research specific? So even in the COVID enclave, we collect data from all kinds of conditions. The filters are really related to which patient's information come in. So we should get any condition that is coded in the original database that a site is using to send data to N3C. There's no restrictions there.
And if you want to see the data dictionary, it's really the OMOP data model. If you look at the OMOP data model, that's pretty much what the data. So it's labs, it's medications, it's diagnosis, it's procedures, it's demographics, and related kinds of issues as they're rendered and represented in the OMOP data catalog.
There's one question: are there data on SUD? I'm not sure what the SUD acronym is.
Substance use disorder? Is that what you had in mind?
Yeah, I think that's probably for me. Absolutely, we do collect all information, all drugs, all substances. Caveat is that substance abuse is severe substance abuse is often treated in community mental health systems and community substance abuse systems. So obviously those aren't in there. There is a dashboard that has a methadone, opioid, tobacco use public report that you're also is also available for use, and it just shows that particular is a nice report. It shows different uses of buprenorphine and methadone in the treatment of opioid and tobacco abuse, not treatment for tobacco abuse, but also concomitant tobacco abuse. So yes, absolutely.
Thank you. So CMS data, Medicare or Medicaid or both? It's both. We brought in Medicare data first and cut our teeth on that. And frankly, that was a little simpler in terms of harmonizing it with the OMOP data model from the ResDac sources. But Medicaid data, as many of you know, is sourced on a state-by-state basis. And oddly, the states have their own ideas about how to render and represent data. So that posed a how do we say this, a greater challenge. But the short answer is we now have both.
Great, and I think we had a couple more questions, Brolin. Said you mentioned March 2024 for the data transfer agreements. How about the DURs? So I guess this is what happens to pre-existing DURs.
Pre-existing DURs should roll on at least for a while longer. As I mentioned, the current data transfer agreements are good for 5 years. There's still discussion as to what the exact end date is going to be and how we're going to make that transition. I wish I had a better answer for you at this point. New DURs, I believe we also decided not to allow new DURs as in wholly new projects after that March deadline. Is that right?
It depends upon
right.
whether
Right.
N3C Clinical becomes a tenant or not.
Right.
For new projects. For example, there are ones that are going into study section, and what do we do with them? It's
and Chris and Richard, I think I may have told you this, we're having a I believe on the 24th of January in CATS. It will either be then or very soon afterwards. We are going to ask all the sites, even if they do not allow us to collect new COVID data, that we at least keep the, we extend the DTA, so it would be another document, more years to accommodate a lot of the things that you and Richard are talking about, R01s, et cetera. So, and we have almost a year to get that signed. So that's at least the plan right now. So hopefully that'll go on.
And Jim is asking what he calls a dumb question. What is the meaning of tenant? And Jim, it's that we have a common housing environment, a common framework, if you will, and that is the integration space where the data is put in and is used for dashboarding and benchmarking and related kinds of things. But when it comes to scientific questions, that's when we partition the framework into houses, if you will. I don't know if you remember in New England, both Jim and I went to school in New England. They used to have tenement houses, and there'd be a tenant in each tenement house. So that's the model where we subdivide a slice of the data for a specific scientific question, and that we've chosen to call a tenant. I don't like the name myself. I'm not sure who to blame about that, but there we are.
Yeah, and I think it's really slicing off related research. It's just one abstract organizational level that honestly relates to governance and funding, being able to support the data analytics for a specific type of research. So the tenant would presumably come with some sort of internal funding structure in partnership with NCATS for a defined scope of research with sites opting into that tenant. But within a tenant, you can still have individual research projects. Another question, do you foresee expanding this beyond observational research in the long term? Meaning, could this be used to form a tenant for sites interested in doing more prospective efforts? I'll let you answer that one, Chris.
Well, I think the idea is that you might want to use the aggregated information in a tenant or other area to identify potential cases and perhaps consider them for a clinical trial. How do you do that? The data set that we have in N3C clinical, both and N3C COVID, is completely de-identified. We do use privacy preserving record linkage. However, we do have the option of going back to the contributing sites. Let's pick on Hopkins or pick on Alabama. Why not? Jim's site. And we could ask that site, these are the cases using the N3C identifiers that an investigator is potentially interested in inviting to a clinical trial. And then it becomes a distributed traditional clinical trial where the sites make the decision, A, whether they want to re-identify those patients because they have the capacity to do so, and then B, whether they want to contact them in terms of potential invitation to a prospective study. So the logical possibility exists. It has not been, to my knowledge, undertaken. We did do a validation study where we re-identified patients, particularly for the long COVID phenotype. That wasn't a prospective study, that was a retrospective validation. We did chart review. So we know the machinery and mechanism works. There is the capacity to re-identify patients. And the expectation is that we could do so with clinical trials, given the cooperation and agreement and agency of the contributing organizations.
We had another question asking how the data submission and the projects are going to be funded or are funded.
I think that may fall under me. So that's the $100 million question. COVID, of course, was obviously emergency, and NCATS and there was Cures Act money to support a national enclave and free GPU for not just the CTSA, CTRs, OCHIN, and others. It was for the entire world could use the data set for free. Unfortunately, that's not really a sustainable model for NCATS to pay for GPU research for any disease across the entire world. And so part of what Richard was saying about the tenant is the idea is that we could create these, they're not, I want to use the word, the tenants are a bigger box than a disease. It's it's renal disease, dementias, or neurology. But in those boxes, you could imagine there would be logical partners. For the renal tenant, which I hope we're going to get funded. It is a partnership with NIDDK, HRSA, and the cancer tenant is obviously with NCI. We've invited some other folks in there too. And so one model, other than the educational tenant, which I can come back to in a second, is to find partners like an institute that said, we would like to help support this. And so that helps NCAS defer some of those costs. It's honestly just too much. It's not that we don't want to. Having said that, of course, if we had a tenant and people all wanted to contribute their data for free, we would be more than happy to take it. But that's the problem. It's really, it's not that we don't want to, we just really can't. So we are trying to partner with ARPA-H and the Moonshot and other large groups that are doing this, so they can take on some of that burden, and then we can just continue to basically use the machinery that we're going for. Regarding the educational tenant specifically, that one I'm hoping will remain an open tenant, because it's really synthetic data like Chris and Richard were talking about it, and we think it is a fundamental imperative for the CTSA, CTRs, everybody else, to learn how to do good algorithmic practice, good data science, good real data research. And we have had it's been not eye-opening, that's too strong a word, but working with 30 billion rows of data is not for the faint of heart. There's a lot of ways to make errors, and having a central repository where we can all help each other and teach each other, a teaching spot, we think is just really important. So NCATS has at least is going to try to keep that one open and free for everybody going forward.
So the sort of related question is another one. Do you have an idea of cost for persons interested in leveraging the tenant for grant funding? I'll start and maybe hand it back to you, Ken. One of the funding mechanisms that is considered is charging individual researchers to do that. I personally don't really favor that model, just because of the difficulty in coming up with that magic number as you're asking, which is what is the cost per research project. I think the funding mechanism that is preferred is kind of what Ken has laid out earlier. If you have an R01 coming in to leverage this for grant funding, hopefully it doesn't cost you anything off of your R01, because the parent agency, let's say NIDDK, for example, is already funding the renal tenant. And if you're doing renal research off of NIDDK, then you would be able to join that, and the compute cost would basically be subsidized by NIDDK directly and not come out of your particular R grant budget. What is sort of nebulous at the moment is, what if you're now working on cancer or kidneys? What do we do there? And so with that cliffhanger, kick it back to Ken for any more insight. I don't know if that has been materialized quite yet, or if these are ongoing discussions.
I love the cliffhanger. It's such a positive way to spin it. This has to be driven by the community, and Josh Fessel is leading it's called a Real World Data Task Force with the CTSA PIs and steering committee to see what they want to fund. It is, if they, as we work towards coming up with what do we do with this, it has to be not just NCAT saying we want this, but the community saying bottom up that this is something we'd support, because this is a partnership. And if NCAT said we are going to spend all of our, this would never happen, but we are going to take the money back and spend it on the tenant, of course I would be lynched. That would be bad, or maybe not. But it wouldn't be, that would just not how CTSA or NCATS works. We really want this to be a joint decision. So there are ongoing talks. I just don't have any more information to offer on the cliffhanger.
We had another question, and I had a similar question, but can you share what phenotyping was used to partition into the different tenants, so like renal or cancer, and is that publicly available?
Yes, they're publicly available. And in fact, the exact phenotype algorithm, I think Richard's was the most simple for the renal phenotype. It was basically if you had a creatinine, yes or no. So that's why we had 3 million patients in that particular tenant, because lots of people had a creatinine. And the idea was that was enough information to infer something about renal function. All the phenotypes were published as executable code in the OMOP data model. And I think they were put into the same GitHub site that we put the N3C phenotypes in, N3C COVID phenotypes in as well. So those are public, and indeed they have to be, because the sites need to have an informed decision to exercise their agency of whether they want to execute that phenotype to generate the tenant.
Or rather have us execute the phenotype on their
Correct.
behalf.
Thank you. Thank
Exactly.
you. So they only have to, that's a good point. They only have to execute one phenotype themselves, which is the blood pressure over a 2-year period or weight over a 2-year period. And that would get them into the staging area, so to speak, where we do benchmarking and harmonization. And then the N3C clinical infrastructure on the Palantir environment actually executes the tenant on their behalf when they agree to participate in that particular tenant. But they can see what it is.
I think we've got all of the questions answered, and thank you both for a fantastic talk. Been there for the beginning of the N3C, and this is exciting to see sort of where things are headed in the future. So fingers crossed, and thank you so very much.
Thank you. Happy New Year, everybody.
Thank you.